Hello, good morning, everyone. Thank you very much for the invitation to participate in this plant biosensors session. And I can see already that there is a lot of interest in plant biosensors and microbial biosensors and other types of sensors that we already started discussing in this meeting. And today I'm going to discuss one of the research programs in my lab that focuses on developing nanotechnology-based tools for monitoring plant health. I am associate professor in the Department of Botany and Plant Sciences, but I also do a lot of my work is also related to engineering nanomaterials for the purpose of developing new tools that can help us to address one of the main challenges of this century, is how we're going to feed a growing population. It is predicted that we're going to need about 60% increase in food production and by the year 2050 to feed about 10 billion people. And this is going to cause huge pressure. It's causing already pressure on our natural resources, leading to deforestation, reduction in groundwater. Well, we are experiencing what it seems to be a chronic long-term drought in the west of the United States. And that is also putting more pressure on land use, energy use, and of course with the consequence of greenhouse gases. So my lab focuses on not only on plant nanosensors, but for the topic of this meeting, I will focus on that. We work at different levels of organization in plants, from single organelles like chloroplasts, chlamydomonas, algae, and also plant model systems like the one we discussed just in the previous talk by Alexander Jones, Arabidopsis plants, which is going to be a good part of the focus on my talk as well. And we are also translating our nanotechnology-based tools to crops for the reasons we discussed, to improving food production and also minimize the effect of agriculture on the environment. So these new tools can be used both for understanding how plants function, but also for engineering plants and giving them augmented or new functions. And we do that by taking advantage of the unique capabilities of some of these nanoparticles. Some of them I will not talk about today, but for example, we have done work using nanoparticles that catalytically scavenge reactive oxygen species, and we use them for studying the role of ROS in plants and also for improving their tolerance to stress, or targeted delivery carriers that can carry agrochemicals or small molecules in molecular baskets to specific cell compartments. And today I'll discuss, for the most part, our work on nanosensors that are based on this structure called carbon nanotubes that are only a few nanometers in diameter and have high aspect ratios. They're very long. They have hundreds of nanometers in length, and that you can code them and functionalize them with different molecules to impart them sensing properties, and also for delivery of biomolecules that could be useful for example, for biosensors. And so this is a growing field. It's been actually even growing faster this year, in which many of my colleagues and our lab have been working on these areas of not only nanosensors for monitoring plant health and environment, but we're also working on targeted delivery approaches for agrochemicals like fertilizers and pesticides. And as I will discuss today, genetic engineering tools for targeted delivery of plasmid DNA, and something that could be potentially used for delivering biosensors, something we can discuss with this group in the Q&A session. So, of course, farmers and people monitoring the environment, they are already using tools for remote sensing, like how a crop is performing in a field, and that, but it's mostly based on traits that show phenotypic changes that are related to changes in chlorophyll fluorescence or leaf area or wilting. But so those methods detect, for example, plant health or stress after the stress has started to lead to some detrimental effects. So what I discuss in this Nature Nanotechnology review is that there's a potential for nanosensors to detect stresses early, right at the onset of the stress, because they can translate plant chemical signals such as hydrogen peroxide. We talk about ABA, calcium, nitric oxide, that are associated to the onset of stresses like drought, temperature, salinity, or pathogen stresses like bacteria and fungi. They can convert in the sense and detect those signals and convert it into digital information, such as like optical, radio wave signals, or electric signals that can be measured by our electronic devices, including even our smartphones, or in specialized facilities like phenotyping facilities using hyperspectral imaging cameras or plant screening systems, or for applications like in agriculture using autonomous manned terrestrial vehicles. So these nanosensors could be used for both basic research all the way to from plant phenotyping to precision agriculture. And there's multiple types of sensors, nanosensors, that are discussed in this review if you're interested, but today I will focus mostly on how the optical nanosensors, how we can use carbon nanotubes for converting, translating the chemical based signal of plants to trigger and coordinate stress responses into optical signals that can be captured by cameras. And I also will do a brief discussion of how we could potentially deliver plasmid DNA encoding biosensors to specific cell compartments in plants for biosensing applications. So I started working in this field as a postdoc at MIT, and in that work, I demonstrated that you can turn a plant, for example, into a detection device for chemicals, hazardous chemicals in the environment, particularly in the groundwater. In this paper, and we show that like how, for example, spinach plants can take up molecules that are from the nitroaromatics family, which is the family of explosives, and take them up by the transpiration, natural transpiration process into the leaves, where the nanosensors can detect the presence of these molecules and modulate their fluorescence intensity, which in turn can be recorded by a camera in the lab, or a camera similar to the one in your smartphone, and with that connect to the cloud and communicate the presence of these hazardous molecules. And I will show you a video of how we do this detection using two types of sensors, like one sensor that is responsive to the analyte of interest, the explosive molecule, and another one that is non-responsive, which provides like ratiometric type of approach in which you can control for factors that are changing the fluorescence of the sensor independent of the presence of the analyte. And using this similar approach, like we have done in my lab, develop other tools for sensing and detecting other types of important biomolecules, such as glucose. So typically, like tools like people use now and to measure photosynthesis rely on or carbon and dioxide assimilation that rely on like measuring carbon dioxide exchange. But in this case, we develop nanomaterials that can have a high affinity to glucose, which is one of the byproducts of the photosynthesis in the chloroplast. And when those glucose molecules interact with these fluorescent nanoparticles that we embed in Arabidopsis plant leaves, they modulate the fluorescence intensity by aggregating and quenching their fluorescence, something that can be measured again with like a Raspberry Pi camera system similar to the one that you can have in your smartphone. And so we can image in real time like changes in intensity of fluorescence that are associated with glucose levels in the leaves. So let me show you this video of like how, for example, this technology can work in the lab. This is specifically for plants that could potentially act as hazardous chemical detectors in the environment. This is the case for the nitroaromatics detection, the explosive detection, analytes. So this is a plant spinach plant that has been embedded with nanosensors in its leaves. And here a researcher is adding a model molecule called picric acid. Picric acid is molecule like from a family of nitroaromatics can be transported from the roots up to the leaves, where the picric acid induces a changes in fluorescence intensity that can be measured with this with different types of cameras. And this is what you will see typically is like a change in the decreasing fluorescence intensity. The sensors can be reversible. And when that is detected, because the camera can be connected to a miniaturized computer, a Raspberry Pi, it can send you a signal, a message, communicating the presence of these hazardous chemicals in the environment. So this is one of the examples of how this potential technology could work, and not only for detection of hazardous molecules in the environment, but also potentially for monitoring plant health and other applications. So moving forward to talk about monitoring plant health. So my lab has been working on developing nanosensors that can monitor molecules associated to stress. One of them is hydrogen peroxide, but you can envision developing these carbon nanotube sensors for other types of molecules present in plants, like nitric oxide or other ones in the environment. And so the way, in simple terms of how you can impart selectivity of these nanotubes to analytes is by modifying their coating using different types of materials. It could be polymers, biomolecules like DNA that tune the interactions of the molecules of interest, in this case hydrogen peroxide, with the nanotube surface or the polymer coating, and affecting its fluorescence intensity. Either a sensor could be responding by decreasing its fluorescence intensity or increasing its fluorescence intensity, or as I'll show you in some cases, even changing the wavelength at which the fluorescence is emitted, a wavelength shift. And in this case, for these single wall carbon nanotubes, we're going to call them from now on SWCNT, near infrared fluorescent sensors, we have, they can respond within leaves, again, here in Arabidopsis plants, by reducing their fluorescence intensity or quenching their fluorescence intensity in response to topical application of the hydrogen peroxide to the leaf surface. And you can monitor this in real time by measuring fluorescence changes with a near infrared camera. And this can be performed at like resolutions that in the order of, like, in some cases of milliseconds. So it can be a high temporal resolution. And these sensors could be potentially also taken down to single particle level detection, something that has not been demonstrated in plants yet, but it has been shown in other eukaryotic systems. So we focus on reactive oxygen species such as hydrogen peroxide because it's one of those plant biomolecules that is generated in almost all types of stresses, particularly abiotic stresses like drought and salinity or heat, UV light or high light. They generate reactive oxygen species, and in part of it, like those are reactive oxygen species are used for coordinated stress responses in plants. But when the plants are at high levels of stress, that could also impair growth, development, and yield, because they can go beyond the levels that plants can naturally scavenge, causing damage to lipid membranes or DNA. And so we are using we're using these carbon nanotubes based sensors that I mentioned, and we with a collaboration of Sebastian Cruz and Bochum University, we functionalize them or code them with with biomolecules here, so like such as aptamer DNA and then these hemming molecules that allow them to sense hydrogen peroxide with like a sensitivity within the plant physiological range. So the way the sensors report the presence of hydrogen peroxide is by the fluorescence quenching of the nanotubes in the presence of the hydrogen peroxide. And we can monitor that, again, similarly to how I explained this before, by using a near infrared camera that can detect like fluorescence about 900 nanometers where the sensors have a stronger fluorescent signal, and using laser excitation, preferably a near infrared laser that can cause less background autofluorescence from leaves. And then you can obtain these near infrared images and monitor the changes in fluorescence intensity associated with the hydrogen peroxide levels. So we tested multiple types of carbon nanotubes. They're not just one kind, but in this case, the 6,5 type carbon nanotubes show the best performance and stronger quenching in the presence of hydrogen peroxide at different levels. As an example, I just show you here at 100 micromolar hydrogen peroxide, like you see a change in fluorescence intensity or quenching. And this, but these carbon nanotubes, like they can also come in different types and flavors. And so in this case, this one is enriched with 6,5 type nanotubes that are fluoresce in the near infrared region around 980 nanometers, but also fluorescing these regions under different excitations that go from like 550 to all the way to 700 and so. So these nanosensors can be selective against biomolecules of interest that are associated with a stress. In this case, we tested for the most part, so molecules that were associated to a stress like stress responses like calcium, sucrose, glucose, ABA, as viscose, jasmonic acid, and others. And the response was not, was minimal, was not as strong as like the one we found for the hydrogen peroxide sensors. And so we, encouraged by these results, we tested the sensors in vivo. And for that, we embedded the sensors in Arabidopsis plant leaves. Like we can do so by either using a syringe with a needle, or now we are also developing other tools that, such as adding a droplet of the nanomaterials with a surfactant. And so the particles like can move through different pathways, but I think mainly in this case, they can preferentially move through stomata pores, as we discussed stomata before, these the openings in like leaf lamina where you can significantly move like particles like by of any size by bulk flow. And so when we have the sensors embedded within the leaves, we can monitor the near infrared fluorescence intensity, as I discussed earlier, using a near infrared camera and exciting them with a laser. And so we can monitor that without having hydrogen peroxide, or after adding hydrogen peroxide, or after removal of the hydrogen peroxide. And so doing this type of experiments, we determined that the sensor sensitivity to hydrogen peroxide range somewhere between 10 micromolar to 100 micromolar or so, where you can see that there is like a reduction, a significant reduction in fluorescence intensity, followed by some partial recoveries. The sensors appear to be partially reversible. And to demonstrate that we actually are sensing hydrogen peroxide in vivo, we did an analysis where like we added hydrogen peroxide at 100 micromolar level, and the fluorescence intensity of the sensors here decreased over like this time span of like about an hour, and then partially recover after we remove the hydrogen peroxide. And this response was not as strong when we also added catalase, an enzyme that's been shown to scavenge hydrogen peroxide, indicating that this is the molecule that we are detecting. So with this hydrogen peroxide sensor, we now ask the question, can we use this as a tool to determine monitoring plant health? And so we tested the response of the nanosensors in leaves of Arabidopsis thaliana to different types of stresses like UVB, highlight, pathogen stress, and wounding that should generate reactive oxygen species like hydrogen peroxide. And we found that in the cases of UVB and highlight, there was a strong quenching of fluorescence intensity of the sensor, followed by some minor recovery, not as much recovery in the highlight. However, under like simulated pathogen stress response by adding an FLG22 peptide that induces pathogen stress responses in plants like Arabidopsis, we found a strong response quenching following by recovery. which could be was very interesting for applications of detecting microbial pathogen infections, and it's something that we are continuing pursuing in my lab, and I'll show you another study that we're doing in this space. What was interesting is that for wounding, we didn't find a strong decrease in fluorescence intensity of the sensors, despite that this was something that we were expecting. We did wounding by piercing the leaves like with a needle in multiple spots, like not in the area where we had the sensors, but nearby on the same leaf. So what we did then was we inquired why this could be the case, and so we quantify the leaf hydrogen peroxide concentration with an enzymatic assay to verify the levels of hydrogen peroxide in these leaves. And so what we can see here is that for UVB highlight and this FLG22 induced pathogen response, the concentrations of hydrogen peroxide are within the range that our sensors can detect hydrogen peroxide within 10 to 100 micromolar or so. Whereas like wounding was like borderline very close to the levels of ROS that where the sensors are not as responsive, around 10 micromolar. So that could be an explanation of why we couldn't detect wounding effects or wounding related changes using these sensors. So I mentioned to you that like we have been very interested in developing plant pathogens, nanosensors, and that this previous nanosensor that we developed was for hydrogen peroxide, and it seems like it could be used for that purpose. But perhaps, yeah, another nanosensor that is more tailored to this purpose is it can detect like plant polyphenols. These are these molecules that you can observe here, like tannic acid and others, that are generated in response of like herbivores or microbial pathogens in plants. And with my collaborator, Sebastian Cruz, like we designed, modified the surface of these nanotubes again for detecting the polyphenols. And so we tested like different types of like modifications, like such as coating them with DNA or coating them with pegylated like phospholipids. And the pegylated phospholipids actually showed the most promising decrease in fluorescence intensity, even concentrations around 1 micromolar. So what you can see here in this matrix is that the pegylated lipids, phospholipid-coated single-wall carbon nanotubes or SWCNT have a strongest response or quench in fluorescence intensity here indicated by blue, whereas like other ones like the DNA coded ones, they show like a milder, actually in relative mild increase in fluorescence, like here shown in red. So the study focused on those pegylated phospholipid SWCNT that seem to be more responsive. And so for proving that or assessing that the nanosensors work, there was different types of analyses, like this type of analysis in vitro, extract with leaf extracts from these plants that grow in the Peruvian forest, rainforest called tococa plants. Collaborators like collected plant leaves that were exposed to herbivory and that were not exposed to herbivory. And with that, we could determine that the response of the nanosensors to total polyphenol content in these extracts. And so we both observe a decrease in the fluorescence intensity and also a change in the wavelength shift. So both indicators can be used for determining the presence of these total polyphenols, total polyphenol content, which can range, can be responsive, like from very low concentrations, like up to like perhaps like 500 or 750 micromolar content in polyphenols. This can be detected, as I mentioned, as a wavelength shift. So there was a significant wavelength shift when you compare plants that are control plants that were not exposed to herbivory relative to those with herbivory that had a significant wavelength shift. And another way we tested these sensors was by challenging, like soybean seedlings that were grown in an agar medium. We embedded nanosensors in the agar medium and then challenged them with pathogens. And so we use elicitors and wounding. And when we use the elicitors that induce the plant defense response, like the polyphenol release, we encountered that the sensors were responding as expected with the decreased fluorescence intensity. And so this is this is one of the experiments that you can see here, that where you have a soybean plant embedded in an agar medium. And you can see this is one near infrared fluorescence emission image that is being converted here to type of heat map type of images for visualizing the changes of the sensor fluorescent intensity in response to polyphenols. So when we induce this pathogen response, we see a large increase in a few hours, 4 to 6 hours, of the polyphenol content in near the rhizosphere, all the way up to 24 hours. And this response only happens when the elicitor is added to the to the roots, but not when is only when the roots are wounded, or when there is no wounding at all. And so the changes in fluorescence intensity are significant relative to these controls. There is some reasonable variation here. And what is interesting too is that you can potentially measure the profile of polyphenol release as you get close to the roots. So you can see here in this distance to the roots, measured in millimeters, you see a highest level of polyphenols as you of course get closer to the roots, and in this area of the roots right here. And so this could be a potential tool that can be used for example, in phenotyping screening systems to detect plants that are, or varieties that are more resistant to plant pathogens because they can generate more polyphenols, or for other applications. Now, the carbon nanotubes are great tools, as I mentioned, for doing these studies and research and plant phenotyping, and potentially we could envision using them as plant sentinels in the environment. However, there are concerns about the carbon nanotube toxicity, and so that is something that we're also working in my lab to specifically target those nanotubes to the leaves, and so they don't move to other parts of the plant or outside into or in the environment. But another strategy is to also develop sensors that are made of more silica nanosheets that can be potentially more environmentally friendly and biocompatible. And these silica nanosheets have also a near infrared fluorescence around 900 nanometers, but they are much brighter than the single wall carbon nanotubes that you see here in these epinter tubes compared to the nanosheets. When we look at the same set of samples in the near infrared, you can see that the fluorescence of the single wall carbon nanotubes is significantly lower compared to the nanosheets. It can be an order of magnitude brighter, which can allow the more easily imaging with cameras and not without the use of laser excitation, but only with LED lights that can result in this type of images where you see the fluorescence of the nanosheets is much stronger than that of the nanotube. So there are other materials out there that could be potentially used and tuned for this type of application. So now I want to briefly mention also some work that I'm doing in my lab that I think could be of potential interest to this community working on biosensors. We are developing a tool for the targeted delivery of plasmid DNA, specifically into the chloroplast. We focus most of our research in the chloroplast because it's an organelle that is important for abiotic stress responses, for photosynthesis, but also because it has a prokaryotic-like genome that is more potentially easier to engineer and across diverse multiple plants species. And so we are developing this carbon nanotubes that are coated in plasmid DNA and also with a chloroplast targeting peptide that can guide the plasmid DNA specifically to this plastic genome. And so this plasmid encodes for the green fluorescence protein that we can track by fluorescence microscopy, and also has antibiotic resistant related gene that could also allow us for select what cells were receive this plasmid and potentially incorporate into the genome. But for what we have discovered is that doing this type of approach is that the Arabidopsis plants in which we deliver this plasmid using carbon nanotubes, similar fashion that to the nanosensors that we discussed before, is that we can have expression of the GFP protein in the chloroplast, both with the non-targeted approach, but with more targeting. With targeting peptide, we can significantly increase the number of chloroplasts that are expressing the GFP. In this case, we have an increase from like 40% to 35% to almost 60% of chloroplasts expressing GFP compared. So this is a significant increase. However, when we look at the expression of the GFP in the chloroplast, we found that this is a transient expression, and it seems like the targeting approach is not leading to higher levels of expression of the GFP. So something that we're currently investigating. Okay, so to conclude.
Yeah. Oh, okay, go ahead. I just wanted to mention that we have just probably 1 minute left.
Yeah, I'm about to finish.
Yeah, thank you so much.
To conclude, this take-home message here is this is an emerging field where we are developing optical nanosensors for translating the chemical signals associated to stress responses of plants into digital information that can be recorded by electronic devices. I have shown you some data sets that support the idea that we can monitor plant health in response to stresses like highlight, UV, and pathogens. And these sensors, based on single-wall carbon nanotubes, could detect molecules such as hydrogen peroxide and polyphenols within the plant physiological range. And potentially, we can use the similar nanotechnology platforms for delivering genetically encoded biosensors. And so the goal is to have the tools that can be translated across different plant systems. We've been working on Arabidopsis thaliana, but these tools in theory should be species independent and could be translated to crops. So with that, I just want to thank my team. So we are an interdisciplinary team of plant biologists, engineers, material scientists working on the space of nanotechnology and plant biology. And we're funded by NSF and USDA, and we are supported also by our work by the NSF Center of Sustainable Nanotechnology. And so if you're interested to join us, actually we have numerous now positionings open due to recent grants that have been awarded to my lab. So please feel free to contact me if you have interest in working in this space. Thank you again for the invitation. I look forward to your questions.